So in this video, we are talking about non-essential amino acid. One of the common and simplest form of non-essential amino acid is glycine. So how many non-essential amino acids are there? It's quite simple. It's 10 non-essential amino acids are there, mainly out of 20 amino acid. Rather than adding the newest form of 20 or 2023, we are talking about all the oldest early 20 amino acids. There is glycine is the simplest non-essential amino acid. Simplest non-essential amino acid in the sense which can be made from serine. So from the serine, glycine can be formed using a glycine synthase. From the glycine synthase, serine will be converted to glycine. So that's the glycine is simplest amino acid. Second way, it's optically inactive. It optically inactive means there is no optical isomers for them. Rather than optical isomers, it can't deflect the light. Optical structures are not there for them. So glycine is optically inactive. Next, we are talking about the major uses of glycine. Glycine is mainly used for heme synthesis, purine synthesis, and glutathione and creatine synthesis. So this is a major use of glycine. For the heme synthesis, glycine is how it is used. Glycine, when which is adding with succinyl CoA, the glycine will be converted into glycine with the succinyl CoA will be converted into ALA will be formed by the enzyme called ALA synthase. From the ALA synthase, ALA is formed. This ALA which can be converted by multiple steps to form heme. So it's not a single step, it's multiple steps are there which can be explained in porphyria. When we are learning porphyria, these multiple steps can be explained there. Here, that's just this basic structure, skeleton for that is glycine succinyl CoA. ALA will be from the essence of ALA synthase. ALA can be converted to heme with multiple steps of that. Then purine. Purine, we all know, purine is mainly from the de novo synthesis. So from the de novo synthesis, what is the role of from the de novo is it's huge complex system from which purine is formed. What's the role of glycine? Glycine is simple. That's if this is a purine, the major part of glycine is here in this the yellow covered area. That is the fourth carbon, fifth carbon, and the seventh nitrogen. Seventh position nitrogen, not seven. Seventh nitrogen, which is provided by the glycine. That's a role of glycine in the purine synthesis. Purine synthesis, it's a de novo synthesis, little complex. It's not that important, but in the sense we should know where the glycine is acting in the purine structure. The glycine is acting the fourth, fifth, and seventh. So 4 and 5 are carbon items. 7th is a nitrogen item. So what's the use of here the glutathione and creatinine? That's the next part. We all know glutathione and creatinine. Here the code for glutathione is both are trimers. It's 3 amino acids are there. Here glutathione is GCG. Creatinine is GAM. So first G of a glutathione is glutamic acid, C is cysteine, and G is glycine. That's what we are talking about. Creatinine, that is GAM. That is first is glycine, A is arginine, M is methionine. So this is all about the use of glycine and the metabolic products from this glycine. These are anabolic products. Glycine is added up. So these are anabolic products of glycine. That's the total things about glycine as an important and simplest amino acid ever present.